Good afternoon to one and all. I invite Dr. Sonia, Senior Scientist RGCB, to give an introduction about the program.
Welcome. Science Setu program, launched by Department of Biotechnology, provides ample scope for the science students and teaching faculty to get an exposure of the cutting-edge research that is being carried out at various research institutes. Apart from attending training sessions at various research, students and faculty also stand to gain immensely from hands-on experiments and demonstration that are conducted during these virtual visits, as the physical visits and gathering are currently not possible. This will be a continuous program with the virtual tours of laboratories, webinars, and live interactions, whichever is deemed appropriate. Today is the inaugural session of Science Setu. We have students and teachers from 13 DVT star colleges participating in this session. I welcome all of you and I request our director, Professor Chandrabas Narayanan, to inaugurate this program. Welcome, sir.
Thank you. Thank you, Sonia. Thank you, Sonia. There is some echo. Should I log out and log in again?
No, sir. It's okay now.
There is no problem.
Oh, okay.
You can hear me.
There is no echo, right?
No echo, sir.
No echo, sir. No echo.
Okay, great. Thank you. Good afternoon, and I should thank Dr. Sonia, Dr. Santosh Kumar, Dr. Sri Kumar, and our RGCB team for organizing this during a very challenging time all of us are passing through. So it's a pleasure to welcome all of you virtually, and rather I would have loved to have all of you students coming and visiting our labs and seeing in real what is happening in this institute. I would like to just give a little bit update on RGCB, since many of you may or may not have completely known about Rajiv Gandhi Center for Biotechnology. Rajiv Gandhi Center for Biotechnology is one of the Department of Biotechnology institutions, and it is currently one of the most best institute in the country under the Department of Biotechnology, and we are happy to say that it is belonging to Kerala. And we have been established about 31 years back. It's going to be 31 years this year. And we started as a society in 1990, but went on to be adopted by Government of Kerala. Later on, in 2006, we were made into a national institute. That is when we joined the Department of Biotechnology. We have been in the mandate, we have understanding of biology that defines basic mechanisms involving disease processes and its implication for human health. This is one of our basic fundamental research goals. Then we also have a translational science as one of the mandates, where transdisciplinary health science to benefit individuals, clinical and public health decisions making to improve health. This is the second mandate. Then we also are extensively into technology development, turning research into technology innovations and on to business. And finally, we also are supposed to do training and education, developing and retaining a sustainable pipeline of biotechnology professionals across a range of related disciplines, including fundamental science, technology development, translation, policy, and outreach through efforts in education, training, and career development. In our institute, we have basic emphasis on disease biology research. And this is done in 4 different directions. One is basic science, where we look at the physiological and molecular biology research in immortalized cell lines or animal and plant models. Then we also embark on early translational science, where mechanistic research in primary human tissues that has direct relationship to human, animal, and plant diseases. Then we also work on clinical science, research involving clinical interventions such as drug trials, new diagnostics, assessment of diseases, disease markers, and epidemiology of diseases. Then finally, we also work in the technology development here, development of devices and platforms for better diagnosis of the disease and laboratory tools for research. Raji Gandhi Center for Biotechnology has predominantly research done in cancer research. A large number of faculty members are working in this area. We do have cardiovascular and diabetic biology research, pathogen biology research, which is another very large group of scientists working here. We have neuro and reproductive biology happening. Then plant biotechnology. Here we have a great emphasis on spice genomics, etc., which is a big thing for Kerala itself. We also have a transdisciplinary biology research happening, which is basically interdisciplinary biology, which is also being done here. We are one institution which has been in the service and training facility. So we do have a laboratory medicine and molecular diagnostics laboratory, which is doing a lot of diagnostics. And we have, in fact, been closely involved with many of the outbreaks of diseases in Kerala. This laboratory has been in the forefront for doing testing and also accrediting various test diagnostics, which are now available. And we were the pioneers in chikungunya and dengue before COVID. And in COVID times, we have actually extensively done testing. We have done about 1.5 lakh samples already for Kerala. And also, we have been accrediting many of the diagnostic tools which are required for RT-PCR and many other equipments from RGCB under the ICMR mandate. We also have another service which we do is medical laboratory service. This is actually associated with most of the government and PHC of Kerala state. And we have been extensively doing testing for underprivileged people, more or less free of cost, because of the government support which we get for running these tests. We have been doing a very close association with most of the government laboratories. And hence, we have penetrated deeply into Trivandrum and Kollam region, but slowly we are actually expanding to Kochi and other places. So soon we are hoping that we will be covering the whole of Kerala with this medical service. We also have a vaccine efficacy center where we are doing a testing for human papillomavirus. Right now, it is being done. It is very well reached a very important milestone in this year. We have a molecular forensic lab, which actually helps the government of Kerala and judiciary to come to a conclusion about paternity and also wildlife. That is a big assignment we have taken up for Kerala government. We also have a research and diagnostic consultancy platforms available here, which is also a very important part. We have joined with Kerala Startup Mission to have a BioNest, which is created in the Kochi area, which facilitates incubation of many of the startups and also facilitates many companies to get their ideas off the desk. So these are things which RGCB has been doing extensively, and now this is another mandate which has been given to us by Department of Biotechnology, where we are going to be training or even teaching many of the students in these star colleges, which would be helpful in them choosing career in biotechnology. So I am hoping that this particular module, which we have actually created for you as a first prelude to many institutions here jointly present, to understand what is happening today in India. Before we go forward, I just wanted to tell you the achievements which we had due to this pandemic, though most of us believe that we had a very bad year. But I think for a research-wise, India, as well as in RGCB, we have extensively benefited due to this particular pandemic, which is a very paradoxical statement which I am making. But what we have realized is this pandemic has taught us to be self-reliant. We don't depend anymore on outside countries to support us or do things for us. We have developed our own diagnostic kits. We have developed our own testing modules. We have been able to come up to this and take on this pandemic frontal attack. We have been able to do a lot of research in this area. And many of the technologies which came out have also reached a commercialization in this area. And we have been able to succeed in many of these from RGCB itself. So with that, I think I will hand it over back to Sonia and her team for continuing with this program. I would request you all to excuse me because I have another program to attend. So I will actually be going out now, and you will all enjoy the program which has been set up by Sonia and her team. Yeah, thank you. Thank you very much.
Thank you, sir. Now we will have a short video on Rajiv Gandhi Center for Biotechnology.
Rajiv Gandhi Center for Biotechnology, located at Thiruvananthapuram, the capital city of Kerala, is an autonomous institute of Government of India, the Ministry of Science and Technology, Department of Biotechnology. Ever since its inception, RGCB is committed towards academic research, innovation, and service. With a major focus on these three pillars, today RGCB stands apart for translating science into public benefit. We discover to ensure a better tomorrow. The idea is to focus on perceiving disease biology and developing this perception into curative application. The inspiring atmosphere of learning at the Institute coupled with availability of cutting-edge technologies, resulted in myriad discoveries in different fields of research undertaken here. Cancer research is our flagship program. RGCB has done commendable work towards prevention of different types of cancer. We are proud partner of collaborative efforts that designed the first indigenously developed targeted cancer therapy without any side effects. Evidence of our scientists on HPV vaccines has become a WHO recommendation, a major breakthrough in RGCB fought against cervical cancer to save Indian women from getting HPV infection. In our quest to go deeper into what is already explored, the Institute runs several futuristic research programs apart from cancer research, now cardiovascular disease and diabetes biology, pathogen biology, regenerative biology, plant biotechnology and disease biology, interdisciplinary biology, neurobiology, and reproduction biology. These research programs at RGCB led to many discoveries and new findings like demonstrating that gooseberry or unlucky protects heart from aging, discovering how Mycobacterium tuberculosis uses acetylation of histones as a mechanism to suppress immune response. Studies showing that pale tree is the new biofactory for new drug discoveries, interpreting role of pregnancy hormone in breast cancer. In response to fungal disease of black pepper, RGCB documented first leaf transcriptome and discovered proteins that are produced in response to fungal infection. This could go a long way in enhancing the production of black pepper in the state. Identifying the peptide from the skin secretion of an endemic frog species of Western Ghats that could work as a lethal weapon against microbial infection. RGCB is searching for antivirals from medicinal plants of Western Ghats to suppress chikungunya virus replication. Research on two mosquito viral infection, dengue and chikungunya is in an interesting phase. Very soon, the world will see a potent antiviral against these two communicable diseases. Our contribution towards understanding kidney disease also covers schizophrenia, varicose veins, type 2 diabetes mellitus in young adults, developing a formulation against cholera bacteria and role of hormone receptors in pregnancy and tumor development. Our scientists' studies on Zingiberesi species have revealed that species with dual reproduction strategy can survive ecological undulation like global warming. In order to decode biotechnology for the welfare of society, we provide various services. Regional facility for DNA fingerprinting within the campus has been providing DNA fingerprinting services as public service since more than a decade. DNA barcoding of species in wildlife, like wild elephants, to control poaching is another service offered by RFDA. Our laboratory, medicine, and molecular diagnostics laboratory provide finest molecular diagnostic services to counter emerging challenges in public health. Our remarkable achievements stands on the strong foundation of superb infrastructure and core research facilities available 24/7 within its two campuses like mass spectrometry, bioimaging, genomics, and animal research facilities equipped with cutting-edge technologies to facilitate higher research and innovation. RGCB is significantly facilitating development of biotech startups. BioNext, a biotechnology incubator facility at Kochi, assists in development and commercialization of new technologies. It is also home to world-class plug-and-play laboratories. In order to create highly qualified, learned, and trained manpower, RGCB runs one of the best PhD programs available and also offers country's first biotechnology skill development programs. Still, the reward for work well done has given us opportunity to do more. Apart from patents and awards, our alumni placed across the globe are our prized possession. Sky Garden, a unique green initiative of the Institute, provides organic, fresh vegetables to fulfill the requirement of campus kitchen. RGCB, a national hub for biotechnological research and learning, truly an institute par excellence dedicated to the service of humanity to science.
Now, I would like to invite Dr. T.R. Santosh Kumar, Senior Scientist RGCB, for delivering a talk on green fluorescent protein from discovery to application.
Hello, good afternoon. Hello.
Dr. Sir. Yes, sir. Yes, sir.
Manoj.
Yes,
Okay,
sir.
thank you. Okay, thank you. So, good afternoon, all. Basically, I am a cell biologist. I am Santosh Kumar, working in the Cancer Research Division of Raji Gandhi Center for Biotechnology. A cell biologist, as I said. I'm working on diverse aspect of cancer signaling and primarily on drug resistance in cancer. That most of you are aware is a serious problem. But today, for the sake of this audience, I'm not talking about any of my hardcore research in cancer. However, I would like to use this opportunity to familiarize all of you with a great discovery in biology. And that how exactly that discovery further help the scientific community to make more and more key discoveries in diverse aspect of biology as well as medicine. Actually, even if you look carefully, you will find that each and every day the discovery which I am going to tell you is actually giving and revealing a lot of tremendous application in understanding the disease as well as other biology, including the COVID cases. The discovery is actually something called the green fluorescent protein. It is actually a protein which has been discovered from a marine organism called jellyfish. Actually, that most of you, I think today we have here with us mostly biology students or biotechnology students with working on either in plant science or zoology or other aspect of biology or probably chemistry. So they will able to understand and this discovery has implication much better than anybody else. That's what I decided to focus on this actually. And the organism from where this protein has been isolated or discovered is shown in the image. Is a jellyfish which is glowing under a visible light, actually as a green in color. And in order to appreciate the importance of the discovery, and probably I will show you in one video in the next. Please carefully observe this video, what exactly is actually happening. I hope actually everybody, this is actually, see, anybody, is it the video is it visible? Okay.
Yes, sir.
I
Yes,
hope
sir.
this is okay. I hope this is visible. Carefully watch this video. This is actually a cell which is growing under in a dish under controlled conditions and observed under the fluorescent microscope. So as you can see that the cells are changing its color in the nucleus, red, to yellow, then it is green, then it is dividing, again it is reaching to red. Hello? Okay. Again it is reaching red and then green and it's oscillating. So what you have seen, probably most of you might have perceived this actually the most wonderful signaling inside the cell, that is cell cycle status. And this is actually one of the great discoveries in biology, because what you have seen is that the cells are actually starting from G1 phase, entering into S phase, then it is reaching to the G2 phase actually. As and when this happens, the cells in the G1 are showing nucleus red in color. Hello? Hello, Rashiran? Hello?
Yes, sir. Yes, sir. Proceed, sir. It's clear.
Okay. In between there also. So again, the cells, actually one of the wonderful events of cells signaling is seen over here. Quite long time, people were wondered, is it possible to see the cell cycle progression inside the live cell without any perturbation? So a lot of work has been already done. And subsequently in 2008, a great scientist called Atsushi Miyawaki used the green fluorescent protein to trace each and every cell cycle phase using different color, using the green fluorescent protein based approaches. So I will explain that a little bit later. And this is possible simply because of the discovery of the green fluorescent protein actually. Now we will start how exactly the fluorescent protein discovery originated. It's actually, as I mentioned, especially those students belonging to zoology will be knowing very well about the life cycle of a jellyfish, Aquaria victoria, and also its structure. And this organism, this GFP is basically inherently present and produced by this organism. And this is actually a first bioluminescent protein discovered is as early as 1960s. You know that this is actually an inherently glowing protein when it's exposed to the UV light. And all this started, the discovery process all started a long time back actually, when one of the great Japan scientists was fascinated with the observation that the jellyfish glows under light actually. So he was so fascinated at that time when in the fisherman community, people usually used to collect this jellyfish from the shore, and they will used to make a scotch out of it, and they used to paste on the walking stick, and then in the night they will use this as a torch actually, because it glows actually. So he was so fascinated by this observation actually. So he started asking the question, what is actually contributing the glow to the organism? So he used to go to the shore, he used to collect a large quantity of the organism, used to bring to the lab and cut. And then subsequently in 1960-61, he isolated a protein from the organism. And then he showed that once you isolate the protein, that itself will glow under the UV light actually. So it is actually the first inherently fluorescing protein from a marine organism actually. So this is actually the image of Shumomida was actually keeping a bottle of protein, which is showing in a green color in the visible light actually, that is shown actually. Now, in fact, the real application of a green fluorescent protein for a biologist was actually first perceived by Douglas Prasher, who at the time actually he was working on a different model organism in development biology. When he heard that actually green fluorescent protein is actually a protein which inherently fluoresces, an idea got sparked that he thought that the GFP could be used as a reporter protein once it's actually made inside a cell actually. That was a time actually, 80s, 87, molecular cloning was actually taking its pace, and most of the people were actually, especially people working on the development biology, was very keen on understanding what were the key proteins and genes which regulate the development process actually. One of the problem they have at that time was that each and every time, if you wanted to trace an importance of a protein in the development biology, you have to kill the organism at specific stages of development, and they have to, stain with a particular antibody, then you have to see under the microscope. But when Prasha heard that, actually, Shimoga developed a protein from a marine organism which spontaneously shows fluorescence. He thought that it can be used as actually a tracer or reporter inside the cell actually. And then again, and he keeping this in mind, because it's basically it's a protein, so there must be a gene for it. And they using gene cloning approach, they cloned the green fluorescent protein gene from the marine organism. And subsequently, Martin Chalfie, basically, he from Columbia University, he was also primarily a development biologist as well as a cell death signaling person who was working on the C. elegans as a model to understand the developmental process. He was also faced with this problem. Is it possible to see a protein expression or gene expression inside the marine in the model organism? So since he got the DNA clone from the Prasha, then he decided to express with a promoter specifically for the elegans, and he showed that once the gene is expressed with a promoter specific for that organism, when the gene is expressed and protein is expressed, you will be able to see that expression inside the organism. So that means they revealed that green fluorescent protein clone or gene can be expressed inside. And this is actually a plot showing the expression of a green fluorescent protein on bacteria E. coli. And then came another pioneering scientist called Roger Chen from University of California. He was a professor of biochemistry department. And he basically was working at actually on cancer signaling in diverse neuromuscular signaling events. And he decided to use this green fluorescent protein and to use as a tracer for signaling events such as calcium as well as ATP signaling. And he extensively worked on the green fluorescent protein by mutating particular amino acids and then created a significant number of fluorescent protein with the different spectral colors. And with this, actually, he developed large number of spectral variants of GFP. And the one of the bacteria plate where different colonies with the different colors cDNA of GFP proteins are expressed is shown over here. As you can see there, some are green, some are yellow, some are cyan, and some are red actually. All these were actually the variants developed by Roger Chen by site-directed mutagenesis of original green fluorescent protein. Now, the question being, biology students, probably you will ask actually, what is actually the implication of an inherent fluorescent protein inside the organism? Basically, that you might have basically studied in your textbook that they have this bioluminescence itself has inherent properties for the survival of the organism where it is present. For example, attracting mates and getting food, and also this is being used by self-defense mechanism against the predators. That is not more important here at this time point. And now the more important here is that now we have a fluorescent protein and we have a gene specifically code for the green fluorescent protein. And also remember, once it is expressed inside a heterologous organism such as E. coli or mice, or any organism or Drosophila, they inherently show its fluorescence when you excite with a specific wavelength of light. More importantly, now with the advent of the site-directed mutagenesis, we have a significant number of fluorescent proteins and genes coding for it, so that you will be able to study and visualize different proteins inside the organism as well as cells with the high spatial temporal relations actually. And very important thing you have to remember is that once the protein or the cDNA is tagged with a a specific gene which are of your interest is expressed inside the cells, that expression or presence of the particular protein is reported by its fluorescence, actually under a fluorescent microscope. If, for example, if you are targeting a protein which is supposed to go to the mitochondria, when you look under the microscope, it will show actually blue in color. This is a blue fluorescent protein in mitochondria. This is actually a cyan fluorescent protein which is expressed at the actin, and this is actually a green fluorescent protein, tubulin, and then Golgi specific protein is expressed here, and vingulin is actually expressed with the Kusabira orange, and there is an M-cherry protein which is expressed in the nuclear protein called histone. So any of the protein, if you wanted to visualize inside the live cells, you take that gene of that and fuse with the specific fluorescent protein which that can be used along with this. And then once it is done, once the gene is expressed and integrated to the cell, this is expressed and the protein is produced. And once the protein is produced, immediately you will be able to see is present under fluorescent microscope. That is the direct beauty of this discovery. And now, these are some more images of green fluorescent protein, expressed with the different tags, which will take you to different organelles. Now, this is actually the palette of color is currently available, and remember the number is not ending. The fluorescent different diverse fluorescent proteins are actually every day is discovered with the different spectral characteristics, and this is the original fluorescent spectral variance is shown over here. And now the implication of this discovery, actually I will explain with the coming slides, so that you will be able to understand how exactly the simple observation and the discovery has actually significantly helped the whole area of biology as well as medicine significantly in the last few years to understand so much of important signaling involved in the diseases. Now, because of that great discovery in 2008, the three scientists who contributed significantly for the discovery process, Osama Shimomura, Martin Chalfie, and Roger Chen, received the Nobel Prize for Chemistry. Now, the discovery of a fluorescent protein is not ending with the green fluorescent protein, as I have mentioned. Subsequently, a lot of investigators came into the field, and large number of marine organisms were extensively exploited for discovery of novel fluorescent protein. And nowadays, we have a good number of fluorescent proteins with the different spectral colors, including the fluorescent protein in the IR region. Actually, this is actually a fluorescent red color fluorescent protein isolated from a sea anemone called the Discosoma species, and similar to that, every month actually you will find discovery of new spectral variants from different organisms. What is actually the importance of this? For example, some of the visible green fluorescent protein as well as RFP red fluorescent protein as such cannot be used for expressing in the bigger organisms such as rat, mice, because they have significant actually light diffraction. So more and more fluorescent proteins with the IR region of excitation are more powerful to image whole animal imaging actually. So these are actually day by day, the numbers are increasing actually. Now coming to some of the advanced application of the green fluorescent protein. Why they received Nobel Prize for the great discovery. Now, this is actually a typical example. Always we know that the structure and functions of all proteins are governed by, all cells are governed by proteins produced by the specific genes. And the proteins, hello? Okay. Proteins, once it is produced, they have diverse functions inside the cells actually. So what are they doing actually? They have to diffuse, they have to mobile, they have to move from one compartment to another. And since actually this is a green fluorescent protein, it can once expressed inside the cell with a particular protein of interest, they will retain the exact biological property of that protein, and then you are able to see as well. And this is actually one of the protein which is expressed along with the green fluorescent protein. And this is actually an experiment where once it is expressed, they have specifically inactivated one particular area. You see that the red area is actually inactivated by bleaching. Actually, once it is bleached, after some time when you look, actually the proteins are moving to that area. That means that that particular protein is present inside the cells in a highly mobile fashion. So for exact functions of these proteins, they have to be mobile in. And sometimes some signaling will inhibit this mobility. So this is actually an important area in biology. What are the proteins which are mobile? What are the signaling which regulate the mobility of a particular protein? So all these can be very easily identified by this experiment. And similar to that, you might have seen and learned in your textbook that endoplasmic reticulum is a single interconnected organelle spanning from outer nuclear membrane to plasma membrane. How is this knowledge came? Your own textbook knowledge. You know, it's actually a single organelle. How this knowledge came? Because it's inter network-like structures. You simply because, see, the experiment is shown over here. A particular protein present inside the lumen of endoplasmic reticulum is expressed with the green fluorescent protein. And a specific area is actually inactivated or bleached by high energy excitation light. Once continuously it is bleached, you are finding loss of fluorescence from the other network endoplasmic reticulum. Why? This is simply because even though you bleach a specific area, the proteins are mobile from the other regions of the ER, and again continuously it is inactivated because this is a single organelle highly interconnected. So this kind of textbook knowledge about the structures and functions of the organelle actually came from the use of green fluorescent protein-based technologies actually. Then coming to some specific signaling which is of importance to all of you is called apoptosis or programmed cell death inside the cell, or necrosis, a non-specific form of cell death, and cell cycle as I showed, and redox changes. Intracellular ATP. ATP is actually the energy currency of every cell. And is it possible to see this? And growth factor signaling. Every cell requires a growth factor binding, and that is signaling to be propagated inside the cell to grow. Then a lot of signaling are there in autophagy, mitophagy, all these are the organelle self-destruction process. And all these signaling, why it is important is that these are very important in diverse disease process. I'm not talking about those importance of these signaling in the diverse diseases. At the same time, I can tell you about some of them one by one, as and when I will explain the importance of green fluorescent protein in addressing these kind of signaling which I have mentioned. Now, if you look apoptosis actually is a program form of cell death. It involves a lot of signaling. You know, mitochondria basically forms the battleground of cell death, where mitochondria releases a lot of proteins called cytochrome C from the intermembrane space to the cytoplasm. Once this comes out, 16 proteases are activated, and the proteases are actually enzymes which cleave lot of substrates after specific amino acids, so the cell will die actually. So this signaling is nowadays well known actually. In fact, it is known because all the signaling actually, for example, the cytochrome C is released from the mitochondria to cytoplasm, and once it comes in the cytoplasm, how exactly the caspase are activated, all these processes were known because of the significant contribution by several scientists. They have utilized the green fluorescent protein based approaches for that discovery, and even that discovery for which they received Nobel Prize. In fact, they significantly utilize the green fluorescent protein as such for understanding the signal. I just will explain you an example. As I mentioned, how exactly the mitochondria undergo permeabilize. Basically, during cell death, mitochondria will permeabilize. Permeabilize means it will basically undergo, form some pores actually. Once the pores are formed, these proteins are released. And then how exactly it is permeabilized? That is one of the very fundamental event of apoptosis, very, very early event. What exactly happens is that a protein called the BAX undergo conformational activation, and then it moves to the mitochondria. You see the same cell, the BAX is expressed with the green fluorescent protein. When it is induced to undergo apoptosis using a stimuli, you see after some point of time, they are actually forming clusters under the mitochondria. So you are actually seeing the event of permeabilization of mitochondria using this approach. See, this is the cytoplasmic protein. After some time, it is undergoing and translocating to the mitochondria, forming pores. Through this pores, the cytochrome C will come out. And similar to that, another event actually, I don't know, you will be knowing a disease called Parkinson disease actually. Parkinson disease is actually mostly damaged mitochondria accumulate in the brain actually. Most often, a particular gene called Parkin is mutated. What is the role of Parkin? Parkin actually is a safe guardian of the mitochondria. Whenever there is a mitochondria is damaged, the Parkin will go and bind to the damaged mitochondria and they will eliminate the mitochondria and degrade actually. If this process is not there, damaged mitochondria will accumulate. So when the Parkin is expressed with the GFP, the mitochondria is shown in with red. I have added one drug actually called which will induce mitochondrial damage. You see the image over a period of time. Very carefully look once again. Is under live image. And see the Parkin actually going and binding to the red color mitochondria. After that, the whole mitochondria, as and when it binds to the mitochondria is degraded. See, this actually seeing is believing. So an important function of a disease-causing protein, you are able to see with the use of green fluorescent protein and its variant, red fluorescent protein. The red fluorescent protein is basically a mitochondria. Now let's see another event. As I mentioned, when cells undergo the cell death, caspases are activated. Caspases are cysteine proteases. Once they activated, they will specifically cleave any protein with a specific amino acid called DVD. So what exactly Chen did is that he expressed two fluorescent protein, one cyan, another yellow. In between, he expressed the enzyme specific amino acid linker, basically is cleavage site. So that once that enzyme is not activated, when you excite the CFP, this acts as a donor, and they will donate the fluorescent, the energy to the venous acceptor, so that you will get emission from the acceptor. But when once it is cleaved, you will find the loss of a FRET, and so that you will be able to see that you will see the loss of fluorescent resonance energy transfer, so that they will go back. So that means this is a cell system. After a drug treatment, the enzyme is activated, it is cleaved. As and when it is cleaved, you are finding loss of that energy transfer, so that more fluorescent from the blue is coming. So you are, with this technique, you are able to see the actual enzymatic activation inside the cell as and when it happens using the FRET. So these are the beauty. And similar to that, what I have shown in the initial video is, I told you that was a cell cycle. That discovery was made by Yasushi Miyawaki. He used two fluorescent protein. One is actually red in color, another is green color, to mark two proteins which are expressed, one in the G1, another in the G2. So they oscillate as and when cell moves to the one phase to another phase. So this you have seen actually. The beauty and wonderful progression of cell cycle you have seen in the first image. He used these two proteins tagged with the two different colors, so that when cell moves to another phase, they will change color in the nucleus. Similar to that, and this is how it looks. Similar to that, if the same cell cycle probe you couple with the cell death indicator. Now I will show you a video. This is actually you are able to see the cell cycle in the caspase activation in the cytoplasm. It is not treated with any drug. If they are growing, the nucleus is changing in color because the cells are progressing from G1, G2, and G2. And when the same cells is treated with a drug, as you see that, you can see the cell cycle in the nucleus red, and cells are changing the color to blue because caspase activation happened, so they are changing its color to bluish because of the loss of red. And what is the beauty of this? You can use, because caspase activation or enzymatic activation is one of the key to kill cancer cells actually. So you express this in the cancer cells. So when you treat with a drug, you will be able to see whether the drug is killing the cell or inducing cell cyclisation. This is actually a big tumor grown on the culture. It's a 3D culture of a big tumor. Then the red in color are actually nucleus in the G1 phase. The green in color are actually the cell cycle stage. You will see a hollow in the middle because a tumor, when it grows in the in vitro conditions, always create a hypoxic region in the core. And that is why you will find a hole in the middle actually. This is actually a confocal C-section. And so basically, that's okay. These are some of the images. And the beauty of, and as well as application of this technology is that we wanted to discover drug killing the cancer cells actually. So use this approach. You add the drug. If they are killed by caspase activation, you will be able to see the color change as you can see from here in the ratio images. So you don't have to do anything. Similar to that, using this system, you can develop animal models. You can develop a tumor inside the animal using the fluorescent protein, and you can use optical imaging approaches to visualize the tumor. You see a tumor which is metastasis to multiple organs in a mice can be seen using the IVs, using an optical imaging approach. This is the beauty of this. Similar to that, a lot of signaling inside the cancer cells or inside the normal cells can be visualized using the powerful approach of green fluorescent based technique. Here, Chen developed an approach for calcium imaging using a cyan fluorescent protein and YFP that will change its color as and when calcium bind to this protein. And similar to that, this is actually the calcium probe is expressed at the mitochondria. One of the beauty of this is that any probe which you develop with the fluorescent based technique, it can be targeted to any organelle, either mitochondria, endoplasmic reticulum, or nucleus. Once it is done, you will be able to visualize the signaling happening at that organelle with the high spatial resolution under the microscope. This is actually a mitochondrial calcium signaling done over a period of time. As you see, initially there is no change. After some point of time, a calcium spark happens, it changes color. And similar to that, redox changes can be analyzed in the live cell using this approach. And this is actually, as you see, a compound called arsenic, which induces significant oxidation damage to mitochondria. As and when it happens, this live cell imaging, they are changing its color. This is becoming red in color because it's completely oxidized. Similar to ATP, ATP is actually a currency of the cells. So a fluorescent protein has been engineered to sense the ATP quantity inside the cells. As and when ATP is depleted, this is the controlled bluish color, and ATP is depleted, as see the cells are changing its color actually. So these are in real-time mode. And similar to that, a lot of pH-specific fluorescent proteins has been discovered recently. The beauty of that is that inside the cells, you will be able to find the organelles when are degraded in the lysosome. Lysosomes are actually highly acidic nature. So any organelle which are degraded in the lysosome, they will change its color because of the pH sensitivity. As been shown over here, the mitochondria is changing its color significantly when it is delivered to lysosomes actually. And this is actually, it can be done in three-dimensional models. As you see, a 3D tumor model which is growing in the culture. As and when it grows beyond a level, as you see, they are becoming significantly mitophagic because there is an acidity which is generated because of the hypoxic core that can be very well imaged by this by use of this pH-sensitive green fluorescent protein. And now I will stop the talk by showing two more videos. This very carefully look. This is actually a cell which is treated with an apoptosis inducer. It is changing its color because of the loss of red and becoming blue. And mitochondria, which is expressed with the mitochondria DS red, still you can see as red in color actually. The bluish color, after bluish color, it is actually it is lost, and mitochondria still it is actually retained because it is stable. It is a stable protein actually. And so here you are looking the real-time visualization of apoptosis. Now, just okay, that protein. Now again, why you have seen the diverse application of green fluorescent protein and in multiple fields. This is actually a time of COVID era. Even for COVID biology, COVID understanding, extensively the green fluorescent protein has helped. As you see, they are inside a cell. We have expressed that ACE2. All of you know that human ACE2 is the receptor for SARS-CoV-2. That receptor is expressed with green in color. So 3 cells expressed with the green in color. And then one of the binding partner from the virus is actually RBD domain of SARS S1 domain. The S1 domain is expressed with the here expressed with the red fluorescent protein. And then the protein is added to the cell. You have large number of cells. Only few cells are expressing the ACE2. The red is actually taken up only by the ACE2 expressing cells. The clear visualization and confirmation showing that ACE2 is actually the specific receptor for the viral protein S1. And what is the advantage of this? It is having so much of advantage. I will show in the next image. And the ACE2, this is actually a ACE2 expressing, and the S1 protein in soluble form purified is added to the cell. After some point of time, you find that the ACE2 cells are taking up and by the S protein with red in color. You see, 30 minutes it is increasing, 1 hour it significantly increases. What is basically in the right side, what is shown is that the same S1 is actually incubated with a neutralizing antibody in my patient. And once it is done, this is not binding as you have seen here. So this is what I said. Green fluorescent protein origin is from a marine organism as early as 1960. And for the discovery of this, they received a Nobel Prize. And subsequently, this has been extensively used for diverse application. Even today, more and more challenges when we are faced with in biology, the fluorescent protein-based approaches are helping the biologists to understand the signaling in a very, very precise manner. My effort was to explain you how exactly a simple observation from a marine organism, probably you can say by local people, subsequently culminated in a discovery how exactly is evolved, how exactly is evolving further to help the field of science. And with this, I will stop, and I can take any question if time permits. I think I have done. Manoj? Hello? Hello?
Here.
I hope that I was audible throughout.
Yes, sir.
It's clear.
Yeah, Manoj?
Just clear.
Yes,
Yeah.
sir.
Then fine, actually.
Okay, sir. Thank you, sir, for the
I think
information. If anybody are having some questions, you can post in the chat box so that we can sir can clear the questions.
No questions?
If there are no questions, we will continue with the program.
I think are they in a position to ask question? That's what I don't know whether
Yes,
that
sir.
permission has been given. Okay.
Yes, sir. Yes, sir. they can give the questions in the chat box. So far, no questions have been posted. I think there are no more
I
questions.
hope that everybody was able to hear me without any interruption. That also I don't know.
Yes, sir. It was clear. The session was clear, sir. It was audible.
Okay.
Thank you for the informative session. Thank you, sir. Now I would like to invite Dr. E. Sri Kumar, Senior Scientist RGCP, for the talk on what COVID taught us. Chat box.
Unable to hear the.
Yeah, good afternoon. I hope I am audible.
Yes, sir.
Okay, so I'll straight away start with the talk. So this is essentially on this COVID pandemic that we have been going on, or we have been going through. It is just hoping to give some insight into what are the lessons we have learned through this pandemic. So first of all,
this conference will now be recorded.
So the current COVID situation as on February 28, 2021, that is yesterday. So you can see that around 11.3 million people across the world are affected. And in India, around 1.1 million people are affected in India. And you can see that the peak of the COVID in India is coming down, and maybe we are seeing that the number of cases are going to come down in the days to come, and maybe we will get rid of this infection very soon because of the introduction of a successful vaccine. So what is COVID-19? As most of us have a good awareness about this COVID as a disease. And now you know that it is a new novel coronavirus infection, which started in 2019, maybe by late December. And it is related to the SARS and MERS coronavirus. SARS coronavirus was another virus that caused a smaller outbreak. It never reached to a pandemic level. But we were expecting that the SARS-CoV also will become a pandemic. Fortunately, it got confined to within the China, and also maybe a few cases were reported outside China, but it never went beyond affecting more than 8,000 people and maybe maximum of 800 deaths. MERS is still confined to the Middle East countries, and even though it has a potential to jump to human beings, the transmission rate of this disease has been much lower. So the MERS has never become a pandemic. Now, all these diseases, SARS-CoV-2, SARS, and MERS, they affect the respiratory system. They cause symptoms including fever, cough, and shortness of breath, and can lead to pneumonia in lungs. The mortality between these diseases significantly vary. And among all this, the MERS is the most serious one, which can cause around 35% mortality. That means out of the 100 people who are infected, 35 may die because of MERS. SARS, the mortality is around 9%. But the SARS-CoV-2, we are seeing that it has, technically speaking, it is around 2% mortality. But in India, we are seeing much lesser mortality because of SARS-CoV-2. And the SARS-CoV-2, the original outbreak happened in Wuhan in China and started spreading globally. And as most of you have an idea that it has an animal origin, it's pangolins or those are the animals which are suspected to be the source animals for this SARS coronavirus. And currently we have vaccines available, but no specific antivirals being available to use to treat this disease. Now, the transmission of this disease is also we should know very carefully. And it is spread from person to person, mainly through coughing, sneezing, and talking and breathing. And the droplet transmission is the commonest mode of transmission of this coronavirus. And aerosol route is not a major, or it's not even considered as a major route of transmission. I have shown a picture here. When a person is coughing, there are 3 categories of droplets coming out. The large infectious droplets will fall around 3 feet from that person. And small infectious droplets can go up to some 3 to 5 feet. And the very small droplets, which are infectious, can travel to a greater distance. And in case of COVID-19 or SARS-CoV-2, the large infectious droplets and the small infectious droplets are the major cause of transmission, maybe more than 99%, I would say. And that is why we always insist that a 6 feet distance between 2 individuals have to be maintained for preventing the transmission of the disease. In the case of other respiratory infections which are transmitted through aerosol, the transmission is mainly through through this very small droplets which can travel at a higher distance. For example, common cold, influenza viruses, all those are transmitted through the aerosol route. So even if you stay 6 feet away, these larger, much smaller droplets can reach you, and the disease can be transmitted. So, and other possible routes of transmission include contact. So we have seen that the hand sanitization is an important step in controlling the COVID. So through the contact, through touching contaminated materials and getting this virus on your hands and bringing them into the eyes or nostrils can spread transmission of COVID. Then other possible routes include the fecal route, because this virus can affect the intestinal cells, which also have the ACE2 receptors, like the pulmonary epithelium. So the virus can multiply in the intestine, and it can be excreted through the fecal material. And so one of the possible route of transmission is considered the fecal route. Now, the transmission of the coronavirus, as you can see, the risk of transmission, suppose a person is walking alone, there is a very minimal risk of transmission of getting coronavirus. But as the number of people or the association between 2 individuals increases, the transmission is very high. Sitting together and having food is a very high chance of transmitting the disease. Crowding together, having parties and crowding together in closed rooms, it increases the transmission of the disease extremely high. So the use of face mask is essentially to prevent the droplets from spreading to other persons, and the physical distance coupled with the face mask will significantly reduce the transmission of the disease. Now, what are the symptoms of the COVID? Most of us have been talking about COVID, and I'm sure that many of us are not very clear about the symptoms of the COVID. And definitely one of the common symptoms of COVID is fever. But you can see that there are a number of people who don't develop obvious fever conditions because of the COVID, and depends on the degree of severity of the disease. And many people develop cough, and some people develop shortness of breath. And these symptoms may appear 2 to 14 days after exposure. And in many cases, people only show up with maybe a throat irritation, throat pain, and maybe extreme fatigueness is also seen as symptoms of COVID. It is not necessary that all these symptoms should be compulsorily present in all these individuals who are affected. And the disease is shown in a varying degree of clinical presentation. And the COVID, you can say that it can appear in two forms. I mean, broadly, like mild COVID and severe COVID. In the mild COVID, the incubation period is usually it can vary from 2 to 14 days, but we consider that from the date of onset of the symptoms minus 2 days, like before 2 days, the virus start appearing in the throat secretions, and it can be spread to other individuals. And the virus spreading or the infectiousness is usually for 8 days, around maybe for a week for mild symptomatic individuals. But for severe COVID, it can be prolonged. And the virus detection that we do for diagnosis, you can detect the virus. Suppose you are using the RT-PCR based system, you can detect up to some 2 weeks of time. And in very severe COVID, the detection of the virus can be more than 3 weeks. And usually for the mild COVID symptoms, usually by 2 weeks of the onset of the symptom, the patient is recovered, cured of the virus, and the symptoms become relieved, and the recovery follows. But in very severe case of COVID, maybe after, not in the beginning of the disease, but after a week's time, the pneumonia changes starts appearing, and because of the poor oxygenation or hyperinflammatory phase that the patient enters into, the patient slowly moves to the stage 3 of the disease, and it may begin with a shortness of breath or hypoxia that can be detected by a pulse oximeter. And slowly the patient, the condition of the patient worsens, and maybe the patient will require invasive ventilation, oxygen support, or initially maybe starting with a non-invasive ventilation, subsequently invasive ventilation. And if the aggressive treatment is not taken, done on these patients during this hyperinflammatory phase, this patient may lose their life. And especially if they are having other disease conditions, comorbidities like diabetes or severe cardiovascular diseases, then the complications can be very high. So these are the usual, and more importantly, COVID-19 is it's not, even though we initially thought about this as a pulmonary disease, mostly confined to the viral pneumonia in the respiratory system. Slowly we started understanding that it is a multi-system infection. It can affect initially itself we knew that it can cause gastrointestinal symptoms. It can infect the intestinal system, and also the virus can be shed into the through the fecal matter. But later on, we started identifying that this virus can affect the cardiovascular system. It can affect the neurological system. It can affect the kidneys. It can affect the reproductive system. So it has become a multi-system infection. And in each of these organs, the COVID leaves some impression of its infection. And whenever this patient is having chronic kidney failure or other comorbidities, including the cardiovascular disease, the disease becomes very complicated. And as most of you know, this COVID virus, this is the spiked structure of this virus, and this looks like maybe you can say it's looking like an anaconda kind of a snake. But as the picture shows, it has a complicated behavior within the human body. It has around 16 viral proteins that is being coded by this viral RNA, which is a positive stranded RNA of around 30,000 base pairs, and it codes for a number of proteins. And the major protein that we always talk about is the spike protein that is projected outside, and which also mediates the infectivity of the virus by binding to the ACE2 receptor, human ACE2 receptor. And there have been a number of coronaviruses that we have been, that can affect the humans, including the human coronavirus NL63, OC43-229E. All these usually cause milder disease, maybe a mild upper respiratory tract symptoms, which many of us would have got it maybe we would have never noticed it. But most of the time, this does not lead to a severe disease. But when this first SARS coronavirus started appearing in Hong Kong, this virus started causing pneumoniacal diseases, and we started seeing the seriousness of corona. And as we have been studying this coronavirus for a very long time, the scientists started understanding that this coronavirus has origin from the bats and maybe intermediary mammalian hosts like pangolins. And slowly then we have this MERS coronavirus, which was identified, that camels were identified as the intermediary host for this MERS coronavirus, which is confined to the Middle East countries. And there are a number of closely related coronavirus members in the coronavirus family, the alpha coronavirus, beta coronavirus of different lineages. And these beta coronaviruses, all these coronaviruses have got more or less the same genomic organization. But these proteins, they differ in their receptor binding capacity. You can see a number of different receptors are used by these coronavirus, and most of these coronavirus have got these bats or maybe bandicoots or rats as the initial host, and they have an intermediate host most of the time. It can be species like llamas or maybe cows or pangolins or camels, and these act as the amplifying host for most of these coronavirus. And whenever they come in contact with the other individuals with these receptors, like mostly this secondary hosts are the humans, and whenever they have this chance to spill over to the human infection, it can lead to a human disease. Most of the, as I said earlier, most of the related coronaviruses only cause a mild disease, but the SARS coronavirus, MERS coronavirus, and this COVID-19 cause severe diseases. We still don't know whether these mild coronaviruses can lead to a cross protection for the severe coronaviruses. But what we see is in the Asian countries or South Asian countries, the mortality because of this coronavirus have been comparatively much less compared to what we see in the West. And there are a number of reasons which has been attributed for the low mortality that we see in the Eastern countries. One could be that we have this natural other coronaviruses circulating in this place. Second, maybe it is suspected that some of the tropical diseases like maybe dengue, maybe chikungunya, or maybe some other diseases are inducing cross-protecting antibodies in the system. It is also not confirmed. And also the age structure of the population. So we still see that the Eastern countries are having more younger population than the Western countries. So maybe because of the better healthcare that is available in the developed world, people with the aged population is very high in the developed world, and also the living pattern of the older individuals, especially in old age homes in Western countries, also may be leading to crowding of these older people in localities, and that is also contributing to a rapid spread of the infection and mortality. So there can be any number of reasons for the difference in the mortality that is being seen in the East and the West. But what we see is most of the time the viral lineages that is circulating in the West and the East remains the same. There is a cross spread of these viral strains from what is circulating. Now we have seen the UK variants in many parts of India. We also suspect that the African variant of the virus has been circulating or has been introduced to the country already. So, but even then, we don't see any significant change in the disease pattern or the disease severity. So, all the most of our talk is or the diseases. The coronaviruses that is causing severe disease belong to the beta coronavirus, and there are avian coronavirus in the gamma coronavirus family, and delta coronavirus. All these other related coronavirus do not jump over to the human so far. So the beta coronavirus are the major ones that is causing severe diseases in human. There are a few alpha coronaviruses also. Now, coming to the lessons learned from this pandemic preparedness. See, we even though we have been seeing many outbreaks of infectious diseases happening in the country. For example, in 2006 or 2005, late 2005, we started seeing reappearance of chikungunya in the country. Maybe after a period of around 35 years, this started reappearing in 2005. Then slowly we could see most of the South Indian states got drastically affected by the chikungunya, maybe during the period from 2005 to 2010, and also the western side of the country. Fortunately, in the initial time period, many of the northern states were spared, but slowly the virus started spreading to the northern states also. Delhi had outbreaks of chikungunya. Calcutta, Orissa, and many other states had this. Gujarat had the outbreaks of chikungunya. Then we have this dengue as a problem for a very long time, and all the 4 serotypes of the dengue virus, which has been, it has become endemic in most of the country, and in certain areas it has become hyper endemic with the circulation of all the 4 serotypes of the virus simultaneously, and also causing co-infections of multiple serotypes. So already and also at least in few parts of the country, we had seen outbreaks of Nipah virus, especially in West Bengal. There were cases of Nipah virus maybe a decade back. And in Kerala itself, a few years back, we had an outbreak of Nipah virus. So all these outbreaks of infectious diseases had already sensitized our system. So we already had a built-in scientific capacity and trained manpower to deal with the COVID situation. So even though our modern science, see, in the COVID learning, there are multiple facets that should be discussed. It has a political angle, it has a social and environmental angle, it has an economic angle, and it has a medical and public health angle, and it has a scientific angle. And since my expertise in the other areas are not that much, I will not venture into take that challenge. But maybe I can talk a little bit on the medical and public health aspects, and mostly on the scientific preparedness that we could do, and also that what way it will be helpful in the future. So we already had a very good scientific capacity and trained manpower in the country to deal with the situation. And even the prior sensitization, especially what we could see from the Kerala experience, and I would know that Kerala was the first state in the country to detect the entry of the COVID virus into the Indian subcontinent. And because of our preparedness and our experience with the Nipah virus, our public health system were very trained to do the contact tracing, and also looking for people who are traveling from Wuhan to Kerala, and identifying any of the initial introductions of the disease. So even though the first case of the COVID got detected in the state in late March, at least a month's gap was there, before which the rest of India they could detect the virus. Can others switch off their mic, please? So it is becoming a disturbance in between. So, so we had that prior experience of dealing with the Nipah, which was useful to the public health system in tracing the COVID, and the rest of the country was already, having the trained manpower in terms of research and diagnostics, and also our medical systems were armed to do so. And interestingly, the research and testing infrastructure, ICMR, Indian Council of Medical Research, had already established a number of research centers. I'll come to that in a minute. And during this, when we also during this during the outbreak time, we could enhance our manufacturing capabilities, and novel testing platforms and assays could be developed during this phase. So this has greatly helped our, how we address or how we, tackle this COVID outbreak. So, we need diagnostics, we need to infuse a huge amount of basic research, and we need candidate vaccines, we need candidate therapeutics in tackling such a huge outbreak. And as I was telling, this molecular diagnostic capacity building, since we had the experience of previous outbreaks of many infectious diseases, ICMR was already sensitized, and they had started a large number of state level viral disease diagnostic laboratories. RGCB was essentially a grade 1 laboratory in the first phase of this ICMR laboratories. Later on, that was transferred to the medical college in Trivandrum, and all the medical colleges in the state and in most of the states in the country, we have the viral disease diagnostic laboratories. And these laboratories were ready to take up the challenge of the COVID diagnostics. And RGCB was one of the first laboratories in the state to start a molecular diagnostic lab. We were offering chikungunya virus diagnostics, we were offering dengue diagnostics, H1N1 influenza diagnostics, and hepatitis B and C diagnosis, and around 35 to 40 parameters of diagnosis we were already doing with respect to infectious diseases. And with the onset of COVID, we also joined the diagnostic capacity building, and as our director was mentioning in the beginning, we have already completed around 1.5 lakh cases detection of the molecular diagnosis. And in India, research and diagnostics have been taken very strong support, given very strong support in the control of COVID, and that is very beautifully controlled and managed by the ICMR in a graded way. And now this has been one of our cornerstones or pillars for controlling the COVID in the country. So in terms of capacity building with respect to and also we could, since we are one of the accreditation centers for many of the ICMR approved center for testing and validating this diagnostic kit, we were getting a large number of requests for approving or validating these kits and personal protection equipments, and this has given us a strong advantage in controlling or dealing with the epidemic. So even though as a scientist who is working in virology and especially in the molecular biology field, we were always thinking that only the space programs or those kind of visible science is getting acknowledged in the country. Now in the time of the most critical crisis that faced by the country, our molecular biology scientists, our medical scientists, and the biological scientists, or who were sitting in the laboratory and toiling for so many years, they could show that India can rise up to the opportunity and cater to the need of the country. So it is a very satisfying moment in that way, that our efforts are being paid off. The scientific capacity building that we have established maybe for so many years after independence is getting recognized and acknowledged. And even during all these meetings that we are attending recently, the capacity building done by the Indian scientists are being greatly appreciated, including the production and testing of the vaccine. Even the Covaxin that has been the viral strain was isolated in India by ICMR NIV, and which was transferred to the Bharat Biotech, and they have made a indigenous vaccine which is being widely and equally successfully used, like the Covishield that has been produced by the Serum Institute of India. And even in the vaccine manufacturing front, we are doing extremely well. Now, in the diagnostic front, for the molecular diagnostic testing, we need the RT-PCR based platform. As you see, this is the viral genomic RNA that is being detected in the RT-PCR reverse transcription real-time PCR system. And we can also use antibody-based test, but as the antibody is developed later in the phase of infection, for early detection, we need antigen testing using nucleic acid detection or the spike protein antigen detection. So these are the different types of testing that we can use Suppose you are using a nasopharyngeal swab as for PCR, you can start detecting the virus maybe 1 week back in the beginning of the symptoms. But I mean, we will not know whether if we have a very clear history of exposure to the virus, we can use this RT-PCR based system. And from the onset of symptoms, initial few weeks, it will be high. I mean, the viral load will be very high, and you can very clearly detect it for a very long duration. Similarly, the bronchoalveolar lavage or sputum, that is also a very useful sample. And virus isolation from the respiratory tract, it is not done for routinely. It is only isolation is done only for research purposes because this needs to be done in a biosafety level 3 laboratory, unlike the PCR-based system, which can be done in a BSL-2 facility. And if we want to look into the IgM antibody or IgG antibody, we need to wait for a week's time, and slowly you see that the IgM is the first one that comes up, and it wanes very fast. Then maybe in 3 or 4 weeks time, it comes down to the basal level. But you see that the IgG antibody response that is elucidated, that is retained for a much longer time. And as all of you know, this is the IgG antibody that we want to evoke by a vaccination that will be giving a person a long-term immunity. And this level of IgG antibodies, how long it persists during the period of that high level of IgG antibody persistence, the person is protected from a second infection. Now, in the real-time PCR, we need to have the tissue sample, which needs to be, we need to do the nucleic acid isolation. We need to convert that into a cDNA because this is an RNA virus. Then this cDNA will be amplified by the real-time PCR amplification. And one of the common methods that we use is a TACMAN probe-based approach. It involves two steps. There is an amplification step along with a probe binding and detection, which will increase the specificity of the PCR. So this is a very sensitive test that is being widely used now. And interestingly, the COVID has provoked many companies to develop their own RT-PCR real-time PCR kits. So even though initially the cost of the kit was much higher, even there was a time when the cost per test was around 4,000 rupees, 3,500 to 4,000, which has drastically come down to around 1,000 rupees or 1,200 rupees. So this is one of the tests that is being widely used. And most of these diagnostic tests will look into two areas in the SARS-CoV-2 genome, that is the RNA-dependent RNA polymerase and the envelope protein gene. Two targets are being used. And for the first-line screening, they will use an envelope gene. And in second, usually it is only one more gene, and in some cases, N gene is also used as an additional confirmatory assay. And this test has been done in most of the diagnostic laboratories. And there are a lot of new capacity building has happened in this. The loop-mediated isothermal amplification, which does not need a PCR machine as such and can be accomplished in a normal incubator with a fixed temperature. You don't need a temperature cycling here. And you can see that just based on the development of a color, we can diagnose the amplification. So this is a technology which was being promoted greatly, but maybe because the sensitivity of this particular assay was not very comparable with the real-time PCR, this technology did not take off very easily, and maybe there was some regulatory approval issues or delays that happened, and now still I think this remains mostly in the research laboratories, but not widely used in the field level clinical diagnostic laboratories. And there are lateral flow tests both on antigen detection platform and antibody detection platform. And as you know, the rapid viral antigen tests are being used extensively for initial diagnosis, especially for airport-based screening and also for field level surveillance. This is being used widely, and even now, when the large number of cases have to be handled, sometimes we resort to using this rapid antigen-based kits. And another capacity building that we could do, and this is one of the research activities, as Dr. Santosh was mentioning, we were also into the R&D in the SARS-CoV-2, and this is a pseudovirion-based assay that we are developing in, along with Dr. T.R. Santosh and my laboratory. We are jointly developing the pseudovirion-based assays for screening the antibody response. This is a green fluorescent tagged pseudovirion. So you already have listened to him about the green fluorescent protein, and we are using this as a cell-based assay for looking at the spike proteinase to interaction. So this is a very beautiful assay to look at the neutralization ability of the antibodies produced in patients against an infection or a vaccination. And this is one of the assays that we are using to look at the neutralizing antibody levels that is developed against the infection. Now, in the case of SARS-CoV-2, another major capacity building that we have seen, or another major lesson that we have learned, is we don't need that much time to develop a vaccine. See, that happened because we had prior experience, or everything was ready because of the initial SARS infection or MERS coronavirus infection. We knew what are the candidates to be used, and we knew what are the target proteins. We knew what are the systems that can be developed. That can be fine-tuned to develop that into a SARS-CoV-2 vaccine. So the traditional vaccine development, these are the different phases, the design and exploratory preclinical studies. In SARS-CoV-2, this was already at least done with the SARS-CoV virus. So we have a very clear idea what should we be, what are the proteins to be targeted. So the process development and preclinical toxicology studies, at least the prior experience, helped us to do it very fast. Then the initial new drug discovery, inventional new drug, could be given to the regulatory approval much faster. So usually it's a very long process. So now with the new SARS-CoV-2, the whole time duration for this can be it could have been very easily, we could reduce that to some 10 months to 1.5 years totally. So from the last March to maybe this March, within a year's time, we have already an effective vaccine in the market, and we can also see that large number of individuals have taken up this vaccination. And now today in the newspaper, we were seeing that in healthcare workers who have already taken the vaccine, there is already a reduction in the number of new infections that is happening. So the vaccine has started working, and we can hope that within few months we will see a drastic decline in the number of new SARS-CoV-2 cases. So all these happened because of the capacity building that we could do because of the prior experiences. We learned from the SARS, and now we have been applying this to the SARS-CoV-2. And this is the kind of learning that we should do with respect to the SARS-CoV-2 also, so that in the future epidemics, future pandemics, we can use this in a much faster way. So now we have all the technical systems for vaccine development in place. And coming to the types of vaccines, we have moved from the conventional vaccine. The conventional vaccines are the live attenuated vaccine and the inactivated vaccines. We have moved into the viral vector vaccines, which is the one that we are using for the Covishield, which is a vectored vaccine where a chimpanzee adenovector is being used. It's a replicating viral vector vaccine. Now, we have been talking about this DNA vaccine for a very long time. I still remember maybe 10 years or 15 years back, we had the idea of using a DNA vaccine, but it never materialized or it never came into the clinical mode. But now we have been licensed an RNA vaccine, which is a newer technology. In fact, it is a derivative of the DNA vaccine technology. But this RNA vaccine, this is a new generation technology, has very fast come to the market, and the Moderna is making a new vaccine, which is RNA-based vaccine. And there is a surge in the vaccine field with respect to the new generation vaccines. And also it has been proven that these vaccines are working in human patients. So this has given a significant boost into new generation vaccines. And we hope that in the coming years, many of the existing vaccines like these traditional vaccines may get replaced with the new generation vaccines, which will also address new diseases incorporated into them. So this vaccine field is seeing a tremendous boost because of the COVID and the successful generation of a vaccine in such a short time. And the vaccine pipeline, the duration has significantly come down. The manufacturers are very hopeful that all the new generation and new vaccines that they will make will get a very fast approval, and they don't have to wait for maybe 10 years or 15 years to market a product, so that delays their profit. So that when the new companies were not interested in this making vaccines most of the time, but this will give a new surge to the surge in the future. And this is also good in a way that our vaccine capacity building is the only thing that we can rely on in the case of a future pandemic. And essentially by vaccinating, what we are trying to achieve is a herd immunity. When a majority of the people who are in the community who are immune to a particular disease, even if there are 1 or 2 individuals who are infected, and the non-immune people in the crowd of the immune people are protected from the disease. So this is the concept of the herd immunity, and slowly there are only 2 ways to achieve the herd immunity. It is only one by the natural infection and the second by the vaccination. And we see that slowly our population is getting into a herd immunity, both by way of the natural infection and by vaccination. And that will also tremendously help in eliminating this COVID from our midst. Now, drug discovery and development activity is another major thing that is required in the face of a pandemic. And usually, just like the vaccine development, we need to spend a lot of time in the basic research and the drug discovery platforms before which it can be made into the drug development activity. And the attrition rate in drug discovery is extremely high. We start with a large number of compounds, and in each of these stages, in the drug discovery phase 1, in the preclinical development, in the clinical development, and in the regulatory approval, you see that the number drastically reduces. And finally, to get one compound approved as an antiviral in the market, we need to test maybe millions of compounds in the beginning. So this is not a very fruitful process many times, and that is why most of the companies are interested only to make drugs against chronic diseases or those diseases which are persisting for a very long time or frequently appearing in the population. So many times neglected diseases do not get any sufficient attention. And usually this, just like vaccine approvals, it also takes around 10 to 12 years, to get an approval. So in the face of a pandemic, the easiest method would be to repurpose the existing drugs, which we have successfully, tried in the case of COVID-19. We have used remdesivir, we have used hydroxychloroquine, we have used many other antiviral drugs, to treat the COVID patients, with some of them showing some level of success, but none of them showing 100% success. So we need more and more specific drugs for any new emerging diseases. So we need to have the capacity built for making new drugs, and also a system to evaluate the existing drugs as fast as possible. So in this particular context, we need to think about some kind of molecules like broad spectrum antivirals. Maybe the antivirals itself is a maybe a recent introduction. We were always hearing about the antibacterials, but now we can very, very firmly or very, very boldly talk about the antivirals. When we were in our younger classes, we were only taught that we can treat only bacterial infection. We don't have any drugs against viral infections. Now we have a number of antiviral drugs available in the market, thanks to the development in the modern science. Now we are hoping that apart from a targeted antiviral drug, we can go for broad spectrum antivirals. So from one drug to one bug concept, we are moving to one drug to multiple bugs. So this is a new concept that is coming up. So this one drug to multiple drug also has 2 approaches, which can directly target the virus or which can target the host. This is particularly important for a viral disease because these viruses depend on the host pathways for their survival. So either you can directly target the virus, or you can target the host pathways used by these viruses so that the viruses are not able to replicate well in the system. So for antibacterial drugs, we don't have that kind of a flexibility. But for antiviral drugs, we have that kind of a flexibility, either to target the virus or to target the host. So, this is a new paradigm that is coming up. There are a large number of antiviral drugs which are already approved for certain other diseases, other viral diseases, which can be tried in a broad spectrum way in a repurposing approach. And this is one of the major things, or stimuli, or the COVID has been one of the major stimuli for this kind of a drug development approach. Now, coming to these global pandemics and emerging infections, COVID is not the first pandemic that has faced the human world. That we have seen large number of pandemics, and it is for sure that COVID is not going to be the last pandemic the human race is going to face. There will be more and more pandemics in the years to come, and maybe it will be in this generation, or maybe it will be in the next generation. But it is for sure that COVID will not be the last pandemic that human race is going to face. And there are a number of factors contributing into emergence of these pandemics or infectious agents. It can be changes in the agent, or it can be changes in the host, our behavior, our susceptibility to infection because of the immunosuppression, poverty, social inequality, or we are talking hugely about the climate changes and the changing ecosystems, and the technology, the international travel and commerce, breakdown in the public health measures, and the deterioration in the surveillance systems. So there can be a number of factors that can contribute to the emergence of a new disease. And interestingly, most of these pandemics, when you look at them, maybe 80% of them, 70 to 80% of them, they are coming from animal sources. And most of these pandemics have got a connection with the animal reservoirs. And this is a very, very important thing, because we see, for example, bat has been considered as one of the major animal reservoir. And you can see around 3,000 viruses have been suspected to be present in these bats. And these belong to different families of these viruses. And you see, coronavirus itself, we suspect that around 3,790 viruses are present in the bats. And just imagine, this is only, we are talking about 3 coronaviruses, and see how many more are in waiting. And see, look at the number of new viruses that has been suspected to be rhabdoviruses, the rabies causing viruses, and so many other viruses belong to different groups are suspected to be present in the bat population. And we are talking only about the bats, and we don't know about other wild animals, how many of them are harboring these dangerous pathogens. So we expect that at any time point, these viruses can cross over to the human species. And this is the time when we start very seriously talking about One Health approach, where there are 3 components involved: human health, animal health, and the environment. So this is the combination, or this is the balance between the health of these 3 systems: environmental health, human health, and the animal health. Then only we can have a sustainable life in the face of Earth. And we have been talking about this for long. And this is a very, very emerging concept, and this has been widely talked about in the case of this pandemic outbreak. And in future, this will be one of the major concepts that our take-home message that we will have, or the lessons learned during these outbreaks. See and also, like when you look at the pandemics, many times these pandemics will be very confined in smaller scale, and some of them will be of the medium range. And you can see the COVID-19. See, the number is, this slide is a little older slide. Now we have more than 11 million cases of this COVID. So some of these pandemics will go beyond our control. So we need to be extremely careful with these pandemics. So there are attempts made by many agencies. There have been publications on how India should tackle these pandemics. These are some of the suggestions by the committees. Like, we need to have our own strategic response teams to work on these pandemics. And these are some welcome signs after this COVID. We started seriously. So this never happened with even H1N1 influenza, dengue or chikungunya. We were never worried about this much about these pandemics till we had to face this COVID. So this has become one of the major learning lesson for us, that our capacity building, so far what we have spent in science and technology R&D has not gone waste, and we have a robust capacity building that has been built up, which can be used for the future pandemic. So the future pandemics and emerging viruses are going to be a major public health threat. There are no specific vaccines or treatment against almost all of them because we have not faced them. We don't know what are going to be the pandemics. We have been always talking about the disease X. For the time being, the disease X is the COVID-19, but there can be more disease X that will come and infect our population. And these agents, especially these viruses, when they evolve very fast, they demand strategic approaches in developing diagnostics and antivirals and vaccines. So if we can have broad spectrum acting antivirals or vaccines, or if we have the capacity to develop the diagnostics very fast, we can manage these pandemics very well. And also it will need behavioral changes among ourselves. We should respect the biodiversity. We should conserve the biodiversity. We should refrain from encroaching the wild for our own maybe entertainment purpose, travel and tourism purposes, or maybe commercial benefits. We should stop doing that. And the exotic game meat food habits, we need to think seriously and change them. So all these approaches would be required to stop future pandemics. And we need to be prepared for new pandemics, and the lessons learned will be very valuable in tackling these new pandemics. And thank you. So if you have any questions, then I can try to answer them.
Sir, so we have a question
Yeah.
from Neeta. Sir, I have a question about the vaccine. If the vaccine tries to increase the IgG antibody production in our body, what about people who already have been affected by COVID? Should they take the vaccine?
See, as I said in the beginning, there are two categories of disease. One is a mild disease and one is a severe disease. And those individuals who had a mild disease, they will have the IgG antibodies with them, but the level of IgG antibodies will be comparatively low. So the current recommendation is those people who have had a disease, it is better to take one dose of vaccine, which will give a boosting to the immune system so that the antibody levels will become higher and they will be definitely protected for a future disease, at least for maybe 1 year or 2 years. We are not very clear how long the immunity of the vaccine will sustain, but based on the previous studies on the Hong Kong SARS, we presume that it will be at least for some 2 years. So it is better to take one dose of vaccine, even for those who have had a disease.
Okay, sir. Thank you. I think there are no more questions in the chat box. If there are any queries, you can post the questions. Okay, sir, there are no more questions. Thank you, sir. Thank you for the informative and thought-provoking talk. So we are about to conclude the session. So we would like to thank our director, Professor Chandrabas Narayana, RGCB, and the speakers of today's session, Dr. T.R. Santosh Kumar and Dr. E. Sri Kumar, for their informative talks. And we'd also like to thank all the participants from the various DBT Star colleges. And finally, we would like to thank the instrumentation team for the technical support they have provided. Thank you all once again. Thank you.